Hi, everybody. This is Kara Fitzgerald at New Frontiers in Functional Medicine. I would not be here month in and month out for the past 6 years without the generous support of our sponsors. And I want to tell you about them. And please check out their websites and check their products out. Biotics research. For over 40 years, the foundation of biotics research has been innovation and quality. Their goals remain unchanged: innovative ideas, carefully researched concepts, and product development with advanced analytical and manufacturing techniques. Biotics nutritional products are of superior quality and effectiveness and available exclusively to healthcare professionals. Visit them at bioticsresearch.com. Integrative therapeutics. Integrative therapeutics is focused on inspiring a better lifestyle through better health. By providing meticulously formulated nutritional supplements and valuable resources, Integrative Therapeutics promises to enrich your patients and embolden your practice. Welcome to your Integrative Therapeutics. Find them at integrativepro.com. And finally, I want to give a shout out to my friends over at Rupa Health. They make lab testing easy, fabulous, doable for both you, the clinician, and you, the person being prescribed the lab, the patient. Consider using Rupa as just a super smart solution to all your laboratory needs. Visit them at rupahealth.com.
Hi, everybody. Welcome to New Frontiers in Functional Medicine, where we are interviewing the best minds in functional medicine. And of course, today is no exception. I am thrilled to be bringing Dr. Vittorio Sebastiano to the world of functional medicine. Let me tell you a little bit about who he is, his background. We're going to jump into what I hope to be quite a far-ranging conversation around some of the amazing science that he's up to. Dr. Sebastiano is an associate professor in the Department of Obstetrics and Gynecology at Stanford School of Medicine. His lab has developed a new technology named ERA, that's Epigenetics Reprogramming of Aging, which repurposes the conceptual idea of reprogramming with the goal to promote epigenetic rejuvenation of human adult cells, leaving their identity untouched. This new technology was patented and is being implemented by Turn Biotechnologies, which Dr. Sebastiano co-founded and where he serves as head of research and chair of the scientific advisory board. He's also a member of the Dior Beauty Reverse Aging Scientific Advisory Board. Dr. Sebastiano, welcome to New Frontiers.
Thank you so much for having me. I'm flattered and I look forward to this conversation.
Well, I have had the honor, or just the pleasure of finding your work and listening to you speak. And you've just got lots of really interesting, provocative stuff to not only say, but what you're demonstrating in your lab. I want to just ask you, go back to the beginning and hear what you think aging is.
Well, that's a very difficult question.
Yeah.
I mean, aging is a lot of things that are happening, with time. Right now, so the scientists have been trying to tackle this problem for a very long time. But I think that really right now we are at a kind of an inflection point where, for a number of reasons, most importantly, the fact that technology also is developing, the right technology is developing actually to answer this question. We're at an inflection point where I think there's going to be really a significant, massive revolution in the field of aging. Historically, and I think this has been in part kind of the reason why we have been so quote unquote slow in understanding what aging is, was the fact that scientists have been looking at this problem, through a very narrow lens. So if you think about how in the past we have been thinking about this problem, we have been thinking about, for example, focusing on telomere attrition, which is the erosion of the telomeres in the chromosomes. Some other people have been just only focusing on senescent cells, mitochondrial dysfunction. So they have been tackling this problem through a very narrow space. Yes. What I think is happening right now is really the integration of all these what are now called hallmarks of aging, together with the implementation of cutting edge technology that is really kind of helping us at a very fast pace understanding what aging means. And let me tell you what aging means, or aging is to me, or how I look at the problem.
Yeah. Mm-hmm.
Well, obviously, as I said, it's a very complex phenomenon that still we are far from understanding. But fundamentally, what I believe is happening as we grow old and as we get old is the fact that the epigenetic program of the cells changes with time, and it becomes dysfunctional with time. Maybe we should spend a couple of words in saying, what the epigenetic program is so that
sure.
everybody understands it.
Yes.
So you can think about that. I always use the analogy of a language. So in every language there is an alphabet, which is the letters that we use, right? The alphabet in this case, in the case of the cells, is the sequence of bases that make up the DNA in the cells. Then these letters can be put together into words. Now, in the analogy, the words are the genes, right? The genes that are turned on or turned off in some cells. And so how do you turn on and off these words, these genes? Well, that is thanks to the epigenetic program, which is, in a way, you can think about that as the grammar, as the syntaxis of a language. So it's a set of rules that puts together in a meaningful fashion these words, so that now you have an understandable sentence, for example. Okay, so let's get out of the analogy and let's talk about the cells. So the alphabet is the DNA, the words are the genes, and so cells that are genetically identical, meaning that they have the same genetic code, for example, the cells in the brain or the cells in the liver, they're dramatically different in their function. Why? Thanks to the grammar, thanks to the epigenetic code that turns on in the brain cells a specific set of genes and turns off a specific set of genes. And in the liver, turns on a different set of genes and turns off a different set of genes. So altogether, basically, this brings to kind of the complexity of cell behavior. Now, what happens with time? It happens that this grammar, this set of rules, becomes dysfunctional over time. And so it can happen that in the liver, in the brain, in the muscle, in the skin, genes that are supposed to be on are now silenced, and genes that are supposed to be silenced become active. So to me, I think that the fundamental linchpin, so the fundamental explanation of why we are aging is because this set of rules becomes faulty with time. The good news, though, is that this program can be reset, so can be brought back to a functional, youthful state, because the epigenetic program is malleable, is plastic, is reprogrammable. And that's exactly what we're trying to do.
It's extraordinary. It's extraordinary work that you're doing. I look at changing epigenetics using, diet and lifestyle. And you're, I think, perhaps doing something similar with the volume turned up, and we're going to dive into it. So folks, we'll link to a Hallmarks of Aging paper, actually one of the most recent ones to come out, where the hallmarks are now expanded, and we have a blog on it as well. So of the hallmarks, cellular senescence, mitochondrial changes, nanodysbiosis and chronic inflammation, it's expanded. The driver, in your opinion, are these epigenetic changes that happen?
Yes, they are the driver, in my opinion. And most importantly, I think that for the sake of this discussion and for the sake of what we're trying to do, I think that they are hierarchically dominant over the other hallmarks. What does it mean? It means that if we can since the epigenetic program is the fundamental program that controls a variety of different functions in the cells, if we can reprogram that program, then as a virtue of that, as a consequence of that, we can really impact most, if not all, of the hallmarks of aging. And this is not just theory. we have proved this multiple times across many different cell types. So it is not science fiction, it is reality.
That's awesome. And we, again, people, there's a lot of publications coming out of Dr. Sebastiano's labs, and we'll link to those as well. So you can do your own drill down into the statement, into this really bold statement that he's making. So I guess I have a couple questions. So one question is, and then of course, we're going to spend a lot of time getting into your technology. Of the various epigenetic marks, is there, what are the ones that really stand up as being drivers of the aging phenomena? So that's question one. And then you can fold it into question two. Are we looking at the aging process, the changes to epigenetic programming occurring in a predictable, programmed manner, which enables us to actually measure biological age by looking at DNA methylation patterns, really reliably? Or are we looking at epigenetic drift or damage just due to the wear and tear of life, or both?
Yeah, well, those are two excellent questions. So let me unfold them. So the epigenetic program is a collection of different types of programs in the cells. So, for example, one of the canonical epigenetic feature or epigenetic characteristics of the cells is what is called the methylation of the DNA. So the DNA can be or cannot be methylated in specific regions. And as a function of that, basically the gene expression changes in different cells and with time. But there is a number of other epigenetic features. For example, the way the DNA is three-dimensionally organized in the nucleus. The DNA is also wrapped around some proteins that are called histones that can be modified themselves. And depending on the type of modifications, again, genes can be turned on or off. These histones can also slide on the chromatin, and depending on their position, again, they can impact gene expression. And there is a number of other things that I'm not mentioning right now, but collectively, all these features are referred to as epigenetic program. So that's where basically we are still far from understanding how these different pieces of the puzzle kind of all together come into place to explain why we age. And I can tell you that right now we are kind of at the dawn of this, of this very exciting field. We are, for example, understanding very well how the methylation of the DNA is changing over time. Yeah. And a brilliant example of this, for example, is the fact that there is a number of people and labs and companies that are developing the so-called epigenetic clocks. Yes. So those are ways to quantitatively measure how and where these methylation changes happen with time, both as a predictive tool to measure basically the chronological age. Well, chronological age is not so interesting because we all know, we all are aware of our chronological age, but the biological age is what really matters. So how far are we from our expected chronological age? Are we younger because we have a very good lifestyle, a very good diet, we exercise on a regular basis, we sleep well, and so on and so forth? Or are we older than our chronological age because, again, our lifestyle, sun exposure, stress, and so on and so forth is actually accelerating our biological clock of aging? So those are wonderful tools that are being utilized, as predictors of aging, but also, they're starting to also be used as ways to really measure how a specific intervention, dietary supplementation or any metformin, rapamycin, whatever it is, how that treatment impacts my biological age. So that's kind of on the first to answer your first question. So there is a lot of things to look at. We are starting to understand it, and we have a very, very good understanding right now of the methylation profile. To come to the second question that I forgot now.
Just the whether we're so aging, the predictable change. So some of these changes that are happening on the methylome take, for example, are pretty predictable. but yet we think that aging is also, random, or yeah.
I personally think that it's a combination of both. So there is a deterministic aspect that can really be predicted and can be forecasted in a way. And then there is, on top of that, there is also a random accumulation of epigenetic errors that are driven by external factors, environmental factors. That could be, again, inflammation. Some of those could be genetic. Some of those could be dietary, and so on and so forth. So and that's what makes it exciting but complex also, because it's kind of the integration of two very complex phenomena. One is developmental, so it's already preset in a way in our cells, which can be accelerated or delayed by the environmental factors that are kind of additive. Yeah,
I think that our research, looking at diet and lifestyle interventions, and we have shown some favorable influence on the methyl-M, is probably more in the camp of influencing those environmental exposures. Whereas the work that you're doing is you're diving into the heart of what would be the programmed. I mean, and maybe you're changing both, the environmental, the influence of those environmental exposures, plus this programmed aging phenomena. And in so doing, you're really able to turn back the clock, and we'll dive into that, but I want to, but before we do, I'm curious, just given your background, how you ended up here, as a scientist. So you're teaching at Stanford in obstetrics and gynecology. I imagine this has something to do with the wildly exciting epigenetic time in embryogenesis. But yeah, how did you get here?
Yeah, I think I got here, I have to say, really thanks to the training that I had in a field that seems to be completely disconnected from what I'm doing right now, but which it is not in reality. So I come from, so my training has always been in development and in reproductive biology. So I have always been fascinated by how, for example, sperm and an egg come together and they make, in this magic process, a new form, a new life. Of course, when I was younger, I was looking at that, really from a hardcore developmental biology standpoint. So I was really fascinated about how is it possible that these two specialized cells come together, all of a sudden they become something completely different, and they have all the information to basically develop an entire new body with trillions of cells that are coming from one single cell. So that for me has always been like really mind-boggling, and I've always been excited and fascinated by that. And so I've been studying, how sperm and eggs form, how human development unfolds, and so on. But then, at the end of 2014, I had this kind of epiphany all of a sudden, and I said, "Well, wait a minute," because I became interested, in the aging field, and I said, "Wait a minute, but nature has already figured out how not to age. Why? Well, it's a very simple thing that was really like in front of my eyes, and I didn't see it before. And I started thinking about the fact that aging doesn't get vertically transmitted across generations, right? We're not, as babies, we're not inheriting the age of our parents, right?
Right.
At every generation, babies are born at age 0. And then they age over time. But then, their children are going to be, again, age 0.
Right.
And if you think about that, that's not true only, within every single species, right? If you think about that, in a more philosophical and holistic way, life hasn't aged on our planet from the very first form of life that formed, 3.6 plus billion years ago, all the way up to us. And so that means that nature has already figured out a way to prevent aging, broadly speaking, from happening in life. The question, though, is that it's doing it through a very, very form, which is reproduction, right? Which is, again, going back to where I come from, which is, how egg and sperms come together and then they form a new life, and so on and so forth. And so, to make a long story short, I really realized that nature has already figured it out, and it has figured it out, obviously, from an evolutionary standpoint. What we need to do now is to understand those processes and apply them to cells that are not embryonic, that are not germ cells, but that are the cells that make up our own bodies, that are programmed to die, of course, to age and to die. But we can basically hijack those principles that prevent aging from being vertically transmitted and utilize those principles and apply them to the other cells of the body that do age and do become dysfunctional.
And this is, we know from Waddington, of course, like this is the ultimate epigenetic puzzler. So you must have been really kind of positioned with your background in reproductive biology to be just, of course, you're going to be leaning into epigenetics as the answer to the aging phenomena, because that's what's happening, like cell differentiation. First of all, we're scrubbing clean the information from mom and dad via epigenetics, and then laying down new epigenetic programming.
Exactly. You're actually absolutely nailing it. This wiping out of this epigenetic information happens at a massive scale in the embryos in the first 2 or 3 days of development. And it also happens in germ cells, as we grow older. And that rewiring of the information is required to cancel or to erase the information that is coming from the previous generation and to set up a new information in the new kind of generation. So it's happening massively across the entire genome. Now, the question is, now that we understand how aging happens epigenetically in our genome, we can utilize that information that we are learning from embryos and from germ cells, and we can just apply it to those parts of the genome that are changing and that are becoming older and epigenetically dysfunctional with time. So it's basically a matter of taking an incredibly functioning and performing machinery and just focusing it and repurposing it to very specific parts of the genome that are the parts of the genome that age and become dysfunctional with time.
Let me ask you, I just have to pick your brain. I don't have a reproductive biologist on my podcast that often, but like when we going back to, embryogenesis and scrubbing it clean, I just have 3 questions. One is, are we looking at methylation and demethylation as sitting in the driver's seat of these? I'm curious, just based on my own reading, seems to be. And according to Hoare, we don't lose everything. There's some information retained, and this would be the heritable aspect of the epigenome, right? We can, and we've seen this in like, say, the Dutch hunger winter or the Overcalyx cohort, where there's generations of information that have been transmitted epigenetically. Those I'm curious your thoughts there.
There is definitely a component that is also epigenetically heritable, absolutely. But again, the good thing is that the epigenetic information, even though some of it could be heritable, can still be reprogrammed and be reset in the new generation, in the new cells. And that's because the epigenetic program also is very responsive, and it changes very dynamically in response to the environment. It was actually built for that reason. So the lower organisms don't have an epigenetic program, and that's why they have a lot of capabilities, for example, in generating massive amount, in replicating, for example. But every time there is an environmental change, they cannot respond very, very effectively to those changes. And then, you may have some species that just, vanish because of that, starvation or UV exposure and so on. The epigenetic program was built evolutionary in our cells to be very, very capable of dynamically responding to those environmental changes that can happen very frequently in nature. And so by virtue of that, they are very dynamic, but they can also be reprogrammed, very, very efficiently if we know how to do it. And that's exactly what we're trying to do here.
Do you think that of the epigenetic marks that we've been discussing, I mean, they all are hugely important, but again, methylation and demethylation are pretty front and center during that time.
Yeah, for now, for two reasons. Well, first of all is because methylation is the epigenetic information for which we have the better understanding. Yeah. And so, of course, we can also understand much better the consequences of changing that profile. As the technology grows and as we develop new technology that can actually give us information about the other epigenetic features, of course, those are going to be integrated in the future.
Yes.
And those could also be opportunities actually to develop even new technology that are actually tackling other types of still epigenetic reprogramming technologies, but of course that they tackle different types.
Different angle. Oh, it's ridiculously interesting. Well, I can imagine in 2014, your head was on fire with that epiphany that you could begin to tease away.
I was absolutely on fire. At the same time, every time, unfortunately, that, you have something that has an enormous potential and, nobody had really thought about before, it also comes with a lot of challenges, unfortunately, because there's, at the beginning, you are kind of looked at as a kind of a weirdo who's coming with this, But it's I can say that a few years back after, I can say that the idea and the hypothesis was correct, and I'm actually pleased to see that it's actually exploding. It
is exploding. Yeah. So were you at Stanford at that time? Were you? Yeah, and so you were able to actually begin to tease apart aging as an epigenetic phenomena back in 2014. That would have been the beginning of the development of ERA, what you're now calling ERA. Is that correct?
That is correct.
So can you talk about what ERA is? Now, of course, I know you'll talk about this in a minute, but you're using some kind of a Yamanaka variation. When were the Yamanaka, where does that in the timeline? When were those characterized, and when did we realize we could reverse? But I'm curious about what, just talk about what you did. I guess I'll just leave it open.
So so Yamanaka published his seminal work in 2006 in mouse. And then the year after, in 2007, in human cells. And basically, to simplify what he did, what he showed was that it is possible to take any cell of the body, for example, a skin cell, and by expressing in this cell a set of factors, a set of proteins, you can basically change that skin cell and make it become an embryonic-like cell. So this was like a remarkable finding that, of course, was received the Nobel Prize in 2012. And why is it so important? It's very important because now you can really create from any cell of the body. You can create this group of cells that are embryonic-like. Instead of using embryos to make them, you are using somatic cells, so the cells of the body, to make them. And now these cells are, they can be grown indefinitely, but most importantly, they can now redifferentiated into any cell of the body. So you start from a skin cell, you end up with an embryonic-like cell. These cells are called iPS. And now these cells can become the skin cell you started with, but also any other cell of the body: brain cell, liver cell, skin cell, muscle cells, and so on and so forth. So why is it so important? Because now you can create virtually any organ ex novo. And these organs now are genetically matched with me because I utilized my skin cell to derive the IPS. And so Yamanaka utilized a cocktail of factors. He didn't discover the factors themselves, but he boiled down basically this process through to a subset of 4 factors: OCT4, SOX2, KLF4, and CNIC. So these are the canonical Yamanaka factors that again can make this transition happening. So what he did not realize right away was that the fact that by making the IPS, so these embryonic-like cells, yes, now they can be used to generate any organ in the body, as we just said, but they are also youthful by all means. They are almost age 0. So by doing this process of reprogramming, he was changing basically the identity of the cells. So again, turning a skin cell into an embryonic-like cell. But he was also making an aged cells into a very, very youthful cell, because embryonic cells are youthful by all means. So in 2014, again, thanks to the fact that I was working with iPSCs and among other things, the hypothesis and the question that we had was like, well, maybe these 2 processes that are happening simultaneously, maybe they can be decoupled. So we could achieve rejuvenation without changing the identity of the cells. So what does this mean? It means that maybe I can simply, by tuning and tweaking that process of reprogramming, and we do this, through dosage, duration, and so on, we can start with a skin cell and end up with a skin cell again. So not an embryonic, but a skin cell, but a younger version of the skin cell. So that was exactly the question that we had. So is it possible to tweak and tune the reprogramming process so that we change the age, but we don't change the identity of the cells? And now, almost 10 years after, we can say that, yes, absolutely, it is possible. We can rejuvenate the cells. We still have the same cell type, but it's a younger, we have a younger version of that cell type.
I want to just, exclamation point and just underline the extraordinary achievement of, your epiphany and your, work and diligence in this. It's very, very, very exciting. So Yamanaka took the somatic cells, everybody, and brought them all the way back to pluripotent stem cell, inducible pluripotent stem cell status. So completely de-differentiated. And I know when they were experimenting with sort of creating, growing from these stem cells that, cancer occurred, there was some pretty negative fallout from taking them all the way back there and then attempting to create an organ or I mean, you know better than I do, but I am aware that the fallout was tumorigenesis and some pretty crazy stuff. And so, I mean, it certainly makes sense that you would be thinking, could we actually do this, less extreme? And I think also you guys were the first to demonstrate this in human cells, right? And are you still, I mean, you were the first, but has it been done elsewhere?
Yes, we were the first to show that it works in human cells. There was a paper before us that actually showed it in mice, and it showed it in a genetic model of accelerated aging in mice. But yes, we were the first ones to show it in human cells. And I have to say that we are still probably among the few. There's really very few people that are working with human cells. Most of the work is being conducted in mice and in other animal models. But I am honestly, I'm a big believer, although I often make my life miserable because, of course, working with human cells is much more complicated for a number of reasons. But I actually want to make sure that what I see is directly implemented or potentially, directly implemented or implementable in the clinic. What does this mean? It means that I have seen way too many studies and instances where something phenomenal that was working in animal models then
doesn't translate
in human. And
Wow.
I'm not saying that those studies shouldn't be done. Those studies are absolutely fundamental to understand the biology, the complex biology. But there's plenty of examples of things that work greatly in animal models, and then they don't work in the human context.
This episode is supported by Practice Better, a leading all-in-one practice management platform. Most health and wellness professionals love what they do, but feel overwhelmed with time-consuming administrative tasks. With Practice Better, you can automate tasks like onboarding, scheduling, charting, nutrition planning, and billing to help you better focus on the parts of your practice you love. New Frontiers listeners get 20% off the first 4 months of any paid plan at practicebetter.io with code KF20. Visit the podcast page on drkarafitzgerald.com for more information.
I want to accelerate this process, and I really want to make sure that whatever I'm doing really has an impact for the life of people. And I think really that the only way, in particular, because now the technology allows for that, because we have learned how to culture the human cells, we have learned how to make organoids from human cells. So there is a lot of human models that, of course, are not perfect, but they're much better, for example, than many other animal models. And so what I'm trying to say here is that we decided to take the long path, but we decided really to develop something that could really be concrete and tangible for clinical translation. And because at the end of the day, what I really care about is really make this affordable, accessible, and for everybody. I want this to be a democratized form of medicine that can really impact the life of people, which is what I care about. And so, yes, it's difficult, it's challenging, but I think it's the only way to do it, in my opinion.
Wow, it's just bravo, just extraordinary for sticking to it until you were able to demonstrate it. And you took, I think, skin cells of somebody in their 50s, I believe, and reversed them to equivalent of 20s. Was that what you did? Yeah, talk about what you did. And I just want to circle back for my listeners to say that this was when you demonstrated epigenetics were in fact influencing all of the hallmarks, right? This would have been teasing that out. Okay.
Yeah, no, so we worked with a variety. So we published our work in 2020. And then, we have now expanded dramatically our studies. So we always try, when possible, to work with older cells. So in the paper that we published, we have worked with skin cells. We've worked with other cell types, chondrocytes, which are the cells of the cartilage, the muscle cells. And we have always tried to work with cells that are at least over 65 or coming from people that are at least over 65. In some cases, even 80 or 90 years old.
Wow. Okay.
And again, since then, we have worked with a variety of other cell types: eye cells, muscle cells, skin cells, mesenchymal stem cells.
All in humans.
All humans. Yeah. And again, as much as we can, we try to work with older cells, because again, the aging process that happens when you are 40 or 50 is very different from the age of the aging process that you experience when you are 17, 80 or 90.
Right.
As I think most of the people are very aware of. And so we try, we have been working with very old cells, and we always try to work holistically. Again, to go back to one of the earlier questions, we always try to look holistically at every single hallmark of aging. So every time we do our intervention, we try to see in as many ways as possible to prove that the cells are still the same cells you started with, but useful and more functional. Because again, cells age, and they age across most of the hallmarks of aging. Of course, they're very different in this process depending on the cell type you are dealing with, but they don't age only for one single hallmark. They age across many different hallmarks. And we always consistently see that when we do our approach, so when we bring in our ERA factors, the cells are youthful now, not just because of one hallmark, but because of every single hallmark that has changed in that specific cell type with time, with age, which is, again, speaking about the remarkable power and potential of this technology, and also about the fact that the epigenetic program, again, can be a fundamental kind of tune that we can use and utilize to rejuvenate the cells, comprehensively and holistically.
Really interesting. It's so exciting to me. I mean, it's just beyond. I heard, I was listening to you recently talk about addressing cancer by reversing the biological age of T cells, and then they can get busy in eliminating, the cancer process. I mean, the implications of your work are crazy. So yeah, you're starting with skin, and anybody who's in there, who's getting older is excited about that. I mean, you think about that, sort of from a surface level, but you're doing all of this, you're looking at this in many different skin types, in many different cell types, and the implications of all of them are just kind of breathtaking.
Yeah, because I think that ERA is agnostic when it comes to cell types and tissue types. So in principle, it can be applied to any cell type or any tissue type or any organ type, in the body. Obviously, the challenge is to really to identify, that window of intervention that is crucial for retention of cell identity, but for rejuvenation. And in the case of what you were mentioning, like in that specific instance, so we are dealing with immunotherapy. So which is a technology that is being implemented by by many companies. So it's basically, the immunotherapy is a way to take your own blood cells, the T cells, engineer them so that they can now recognize the tumor and kill it selectively. What happens, and this is a function of age for sure, but it's not necessarily a function of chronological age. What happens is that during this process of isolation, expansion, engineering, and manufacturing of the cells become aged. They become exhausted. That's the term that is used in the field. So they become fatigued. And so once you reinfuse them back into the body, they are not as powerful and as fit and as functional as they should be in killing the cancer. So they have an enormous potential of doing it, but again, they are in a way exhausted in doing so. So this is a perfect example of where ERA can kind of, orthogonally intersect. And since ERA, quote unquote, impacts the aging phenomenon of cells and can bring back the age of a old dysfunctional cell back to a youthful one, it's a perfect example where ERA can really be nicely applied. And so you can make now these exhausted cells youthful and functional again. You can make them younger again. And so now you have, kind of two things that are coming together: their ability to recognize and kill the cancer cell, but also their ability to not be exhausted and aged. And so they can just go and kill the cancer more effectively. And that's exactly what we're working on as well, as a way to really bring this amazing technology to the fruition, not just to the fruition of very few people that use it as a last resort, but as a first in line opportunity that could really wipe out and could really impact
quality of life,
cancer biology, broadly speaking.
I mean, I mean, we could look at potentially prevention, before the need for, this kind of immunotherapy when the cancer is present. And you've talked about that, and I would like to talk about prevention. But before we get there, just understanding a little bit, now that we talked about Yamanaka factors and some of the challenges, and ERA is a different take on that. So if you could distinguish them, I would like that. And I know you certainly it's not like we're going to all go out and take an ERA cocktail today. I mean, there's some challenges there. I mean, cells respond differently, and, the dosage and the duration, and also there's massive variables. And so, talk about that as well. What ERA is and what some of the challenges to translating this to real-time medicine are.
So ERA is basically, again, the biology of ERA is really this idea of reprogramming the epigenetic profile. How do we do it? So basically what we do is to bring inside an H cell a cocktail of mRNAs that then can encode for a specific protein. And these proteins now go into the nucleus of the cell, they reset the epigenetic landscape, and they bring it back to this youthful and functional state. Now, so this is kind of the biology of the process. The real challenge is, how do you bring this cocktail into the cells? And you can think about this in two ways. One way is, well, I first isolate the cells from my body, right? I treat the cells in a petri dish, so in the laboratory, and then I reinfuse the cells back into the body. So this would be the idea behind the immunotherapy that we just discussed. But our vision is to be able to rejuvenate the cells inside the body without prospectively isolating the cells and culturing them outside of the body. But this comes, so the potential here is enormous, because again, we can go into the liver and make it younger. We can go into the brain, hopefully one day, and make it younger. The same is true for every organ, every tissue in the body. But the challenge is that, how do I make sure that I specifically target some cells in that organ or in that tissue? And that's exactly why, Turn not only is working about ERA, so the biology of this process, but is also working on developing new ways to specifically target and deliver this cocktail of mRNAs inside the cells, to make sure that we are targeting only one specific cell type, or maybe more than one, but in a very, very efficient and specific way. And this is a very challenging question. Really challenging. Again, technology now is developing, so I think there's going to be a lot of remarkable findings in the near future, because there is a lot of people that are working on this. So the delivery is really the challenge here. And we are working actually on making that precise and specific. So we're developing our own lipids that can encapsulate and protect the mRNAs until they are outside of the cells, and then being efficient in delivering the cargo, which is again the mRNAs, inside the cells.
And to the correct organ.
Exactly. Yeah. And from a safety standpoint, so you were alluding about, there are always obviously some safety risks associated with this. Yes, absolutely, there are. And the risks come from the fact that if you don't know how to control in time and in dosage the expression of these factors, some of these factors may become quote unquote carcinogenic. But really the key here, and
would that be because of too much dedifferentiation? Like they would just go too far back.
Exactly, you will go too far back, and that's where the delivery technology, together with the understanding of the biology of the cells, kind of come together nicely. Because if you know how to deliver, the cargo in a specific cell type, you already know the biology of that specific cell type, and you know that that cocktail in that amount for that number of days is sufficient to rejuvenate, but not to, lead to cancer. That's basically where the solution is. And that's why we're working hard on that problem to, on the one hand, again, target specific cell types in the body, but also at the same time understanding very nicely and deeply the biology of those cells, so that we can make sure that there is no loss of cell identity happening in this process.
Sergey Young is a friend and colleague of mine who talks about the longevity bridge, and he's a proponent for all the things you mentioned: eating right, getting enough sleep, exercising, all of the things we can do today, as we walk this longevity bridge and we get to, what you're proposing, because there's some time there, I mean, there's some time involved before anybody, when, it's exciting to listen to David Sinclair and to see what he's doing over at Harvard with his various animal models playing around with aging. And I think I may have heard him actually say one time that he might take a Yamanaka cocktail, but it just doesn't, it doesn't make any sense. So, yeah, there's some really clear hurdles, and yet the potential is extraordinary. So rejuvenation, epigenetic rejuvenation, or biological age reversal is going to perhaps look different than, say, Aubrey de Grey, who thinks the first person who's going to live to 1,000 is, that we're going to just, take this cocktail and become 30 years younger. Like, that sounds, listening to you, like it's science fiction, or is it not? I mean, is it not? This is the first step, what you're talking about. Yeah.
Yeah, no, I don't think it's science fiction, honestly. Of course, I think we need to also be a little bit down to earth and not be kind of, too bombastic about our, So what do I mean by this? I think that ERA, first of all, I think that ERA, not alone, but together with many other types of interventions, are actually going to get us there. Are going to get us one day, I don't know exactly when, but are going to definitely get us to a point where we're really going to be able to live, yes, longer lives, but most importantly, healthier lives. Okay, this is what I think is really the point of what we're trying to do. We're not impacting lifespan for the sake of living longer. We are impacting health span for the sake of living longer, healthier lives. Okay, so this is what I think, at least for me, is important. I don't find it so useful to live until 150 years of age if my last 50 are going to be miserable because I am frail, because I have all sorts of issues, right? That's not the point. The point is,
I mean, and that's really what we're seeing now. We're not going to 150, but we're spending, upwards of our final 20 years, many of us, with significant disease, just real quality of life, significant quality of life issues, across the globe.
Yeah. So the whole point, the whole task and goal that we have is really, for now, trying to impact the health span of our current lifespan. And so making sure that, if I am at risk of developing heart disease, just as a function of age, of the function that, I become, older, and maybe I had a horrible lifestyle, maybe I can fix that, or I can prevent that from happening. And of course, I'm going to impact lifespan, for sure, because if I'm not dying, of a heart attack, of course I have a longer expectancy of life. But really, what matters here is to make sure that the quality of life is impacted, and so I don't develop frailty. I am not at risk of pulmonary disease, dementia. And so on and so forth. How are we going to do it? So I think that we're going to do it in many different ways. ERA is definitely one of the ways. And so again, to go back to your question, I don't think that is science fiction in the sense that it's not going to happen. I think it's going to happen at some point. That maybe we're gonna get have a dietary supplementation of some sort that is going to really reboost. Kind of systematically and systemically all our cells in the body. I don't think it's going to be the Yamanaka cocktail. Honestly, I don't think that's not going to be to happen. But I think that's again, ERA together with many senolytics and many other interventions are going to get us to a point where we're going to live significantly longer and significantly healthier lives.
Isn't ERA kind of a next generation Yamanaka? I mean, you invoke we're leaning on the Yamanaka factors in what you're doing.
Well, right now we are using the canonical Yamanaka factors plus two other factors that together with the other four elicit the highest degree of rejuvenation in the shortest amount of time. Again, to go back to the safety issue, if I can make sure that I can treat the cells for just a very small number of times and days, but by using an expanded cocktail, I can actually get a higher degree of rejuvenation with a minimum number of doses. That impacts the efficacy, of course, but it also impacts safety dramatically.
Is that, are some of those other factors helping to just shut it down? Are they?
All the factors are shut down because, again, we are bringing in the mRNAs. So the mRNAs have a half-life, so they hang around in the cells just for 16, 20 hours, not more than that. So you can prolong the expression of these genes if you keep giving these factors to the cells. But if you give them just once for, just one time, they are expressed for 16, 20 hours, and then they're gone. But that time is sufficient for the proteins that they are encoding for to go into the nucleus and reset the epigenetic landscape, okay? So the cocktail that we are using right now is it gives us that opportunity to really get a remarkable rejuvenation in a very short time. Yeah. That doesn't mean that the same cocktail could and should be used for every cell type. We can optimize that cocktail in a cell-specific manner. But in addition to that, we're also discovering other factors that in response to these factors are being turned on or off. And this is going to give us a lot of leverage about, maybe we can optimize this cocktail with other factors, or we could replace some of the current factors in the cocktail with some other factors that maybe are just even more potent in doing so. So there is a lot of discovery also that is happening that is super exciting.
So exciting. It's really interesting. I would just want to say, circling back to my community where we're thinking about nutrition and things like that, you do need, if you're going to rejuvenate someone, you need the ingredients to be able to do the rebuilding. So our world is not going to go away anytime soon, but it would be really fun to partner in helping, something older, have the ingredients to actually enact the information that your work is providing. It's kind of a cool idea for me in my world. Well, you're at clinical trials, right?
Not yet.
But are you near?
We are near, yes. So we are having in the next few days, we're going to actually have our first conversation with the FDA.
Wow.
So this is new territory for the FDA. So I think there's going to be a lot of very, very crucial information exchange that is going to help them understand what this technology is all about, but also help us understand how. From a safety standpoint and from an efficacy standpoint, we should start thinking about the clinical trials. So we're very excited about that because I think this is going to be a very, very interesting and very important conversation with the regulatory agency. And so again, depending also on other things, for example, funding, for example, of course, we're not in a super friendly market environment at the moment, but
it's got to be wildly expensive.
Yes, so but we are very optimistic, and I think that if everything goes smooth, we could really get to a phase 1 clinical trial, which is a safety phase, maybe by early 2025.
Wow. Just super exciting. All right, so let me just circle back to the work that you did in human skin cells and the fact that you're with Dior. I mean, are you thinking about topical delivery? I've heard you talk about microneedling once upon a time. Is that something that may move closer to use in humans? I mean, speak about that.
Yeah, I think so. Definitely. I mean, again, we have to also deal with the fact that there is some other technology that needs to be developed in the meanwhile that is going to complement what we are doing. But speaking about the skin, right now, the way we're thinking about this would be to deliver into the skin for kind of medical associated cosmetic issues to begin with, but maybe one day just, cosmetically, to really bring these factors inside the skin, for example, through microneedling, because that's the technology that it has been developed. It has a lot of history. There is a lot of knowledge about it. So that's the technology that we have available right now for the delivery. or for the route of administration, I should say. But yes, absolutely, we're also working on topical formulations that, you know, would not require any microneedling. It would just be a cream of some sort that could be topically applied, for example, in wound healing. For example,
right.
You know,
or burns.
Burns, exactly. Those are all phenomena or all indications that are severely impacted by aging. So the healing capacity of aged skin is dramatically impaired, okay? So if we could accelerate that process, if we could make sure that, you know, the healing happens much faster, and that also by doing so that the thickness of the skin becomes, you know, thicker, and so, you know, the skin becomes more resistant to pathogens. It becomes more resistant to wear and tear. So that has a lot of repercussions also on the future, right? Because, you know, at that point, the skin becomes more resistant to a variety of inflammation. It becomes more resistant to pathogens. So that means that there is no inflammation happening, and that means that there's no accelerated aging. So, you know, the consequences of this could really be endless.
Where are you at in that journey? Has that moved into any clinical trials?
Not yet, but again, we are actively working on preclinical studies right now, and we are developing, to go back to the challenge of the delivery technology, where we are developing topical formulations that are going to be actually used for the delivery of mRNAs into the skin. Again, it sounds simple. It's not simple at all, because the skin is one of the most complicated tissues in the body.
Interesting.
And it comes with a lot of challenges, you know, from a chemistry standpoint, but we're trying to solve those challenges. And again, this can have a repercussion on a variety of different things, because you can even think about delivering other types of genes in the skin to make it, again, more functional. And you could even at some point start thinking about genetic correction of some skin diseases, for example.
Interesting. Yeah. You have talked about ovarian rejuvenation as well. And this is an area, like, I think of particular interest for you. Speak about that and where you see ERA.
Yeah, so ovarian regeneration and rejuvenation is actually one of the things that is really at my heart, because I think, you know, personally, I want to really impact women's longevity and women's health span. Again, we have to think about the problem of aging from in its complexity. To go back to what I said before, ERA is going to be definitely one of the solutions, but it's not going to be the only solution. So one of the things that, you know, my lab is working on is to really show that we could regenerate and rejuvenate the ovarian tissue through ERA, yes. So by, again, going in with the same cocktail of factors and bringing back the ovarian tissue in time to the point where, you know, again, it's youthful and functional, but also through generating new ovarian tissues ex novo, for example, from iPS cells. Why is this important? For a very simple reason. The entrance in menopause is inversely correlated. The age of entrance in menopause is inversely correlated with the life expectancy and health span. What does it mean? It means that the earlier a woman enters in menopause, the more likely is going to be that the lifespan is going to be shorter, and the health span is going to be impacted, you know, from a cognitive standpoint, and, you know, from a health standpoint.
I mean, just going back to the original conversation about, I'm sorry I interrupted you, but say that, like, about this programmed aging phenomena, like menopause seems sort of like the classic evidence, you know, that something, you know, that this change will push us towards death in a very predictable way.
Exactly. And it's an accelerated aging phenomenon that happens post-menopause.
Yeah.
But in addition to this, I think there is a quite a significant number of women that enter menopause, not when they are, you know, in their late 40s, but they enter menopause when they are in their early 40s, or maybe in their 30s, or in their
yes,
because of premature ovarian failure or premature ovarian insufficiency. So now we have a number of women that are very young that experience accelerated aging because of loss of functionality of the ovarian tissue. So we can make an impact. We could potentially make an impact. And if we could extend the functionality of the ovarian tissue for much longer, we could actually give them an opportunity actually to at least to age in a quote unquote physiological manner, right? And not to accelerate, their process of aging. But again, broadly speaking, you can apply this potentially to any woman, and you could really make a significant impact on the health span. Yeah.
Extraordinary. I mean, that's a key leverage point in women, because what the ovaries do is so impactful globally. I mean, the central nervous system, brain health, heart health, bone density,
exactly,
muscle. I mean, and on and on. It's extraordinary. So starting there could be a really appropriate entry point into bringing ERA. Wow. And having just really a global rejuvenating effect by only needing to address a single organ.
Yeah, and that's actually interesting what you are saying, because again, I don't think, at least in the short term, I don't think that we necessarily need to think about having the long life elixir, that we're going to drink to be younger. Maybe just by working on a number of subsets of tissues, we can have this systemic global effect. But just by doing one simple thing, which is targeting one organ at a time. Yeah.
That's very cool. I want to just, we need to wrap up here, but I have about 20 more questions. But no, I wanted to talk about telomeres. So telomeres are one of the hallmarks, telomere attrition, one of the hallmarks. But what you've seen in your research is that if you're elongating telomeres, you're actually hitting cell dedifferentiation. It's like it would be a bad sign in your work. And I think that you've said that our focus on telomeres may be misdirected. Can you just speak to that? I mean, it's one of the things that comes to mind first and foremost when we're thinking about longevity.
Yeah. So yeah, it's a good question. Let me clarify what I mean. Okay. So for example, there is an existing technology right now that is thinking about, for example, through the mRNAs to bring inside the cells the gene that is responsible for the elongation of the telomeres, as a possible solution to aging. I think that's definitely one of the weapons that we have in our arsenal to tackle the problem of aging. So I don't mean that we shouldn't be looking at that, and that is dangerous on its own. Like for our factors, I think that if you can control that process and you can make sure that you just temporarily elongate the telomeres, but then you don't overdo it, because if you overdo it, then the cells become carcinogenic. I think that that's a splendid idea, and I think that people should be pursuing and following up with that idea. What I mean though is that the telomere attrition is just one of the many different things happening with age, and so it doesn't explain why we age. And telomere attrition is a phenomenon that primarily impacts cells that become senescent, which is again a subset of cells that is only maybe explaining 1% to 5% of the cells that we have in our body as we age. And so solving the problem of telomere attrition is important, but again, is going to affect only a small number of cells and is not going to have such a dramatic impact comprehensively on the process of aging. So that's what I mean.
So there may be a subset of people for whom that would yield, benefit, worthy of the intervention. Exactly.
And to explain why in our case we do not see telomere elongation and why we think it's good for us.
Yeah.
Is because so during the process of reprogramming, the gene that is responsible for elongating the telomere becomes active when you are far away in the process of reprogramming. So when the cells are almost about to become iPS. So the reason why we don't want to see telomere elongation happening in our case is because if we do, that would mean that the cells would have lost their identity and they would become carcinogenic. But that doesn't mean that together with our cocktail, we can bring in again, temporarily, the gene that is responsible for telomere elongation, but just making sure that it is expressed for a very short time and not for much longer, if it makes sense.
Yeah, it does make sense. I guess I'm just not, I don't know that I'm as convinced that that would yield. No, I mean, it's not my field. I'm just like an armchair opinion generator over here. Like it might not, I mean, I'd rather have my ovaries rejuvenated with an Era cocktail. Anyway, although I guess we could do both. It'll be interesting to pay attention to that as that technology, as they actually do telomere elongation and to see what outcomes occur with that. Listen, it was just been a really great conversation. Thank you so much for your time and your ability to explain in really plain language and bring this field alive. And I appreciate your commitment to this work and your passion around making the world a better place for all of us and making it affordable. Thank you.
Yeah. Thank you for having me. And yeah, absolutely. This is really what drives me, what gets me up in the morning. I really hope that this is going to be really something that everybody can utilize, at some point, because I'm really a big believer of democratization of medicine, and I really hope that this is going to be applicable to anybody with no distinction, in terms of who can afford it or not. Yeah. So, and thank you actually for giving me this opportunity, because I think it's important that the information gets out there and people are aware of it.
Yeah, absolutely. All right. Well, Dr. Sebastiano, welcome again. Thank you for coming to New Frontiers.
Thank you.
Hi, New Frontiers listeners. So when I finished my conversation with Dr. Sebastiano, I realized I had a lot more questions for him. I was grateful he was willing to hop on a call with me and chat through these additional thoughts. So what follows is a deeper drill down into the science, but it's incredibly interesting. For example, why did he choose to focus his research using human cell lines and human tissue rather than an animal model? Is this more translatable into human application? The answer, he believes, is yes, and he's nearing FDA approval to start clinical trials. So listen to that. The other huge question that we covered, among other things, was whether or not epigenetics is the root of aging, or is focusing on epigenetic changes, manipulating DNA methylation and so forth, the most effective entry point into reversing aging. So listen to what he has to say about this. I think that you'll be incredibly interested in it. I'm grateful for his time. And really, this is one of my favorite podcasts of the year. Thanks so much. So you're a part of the Biomarker of Aging Consortium, which I think I'm excited about. You're going to have a meeting, I think, the first meeting, if I'm not mistaken, in December at the Buck Institute. Can you, are there, I know you're obviously DNA methylation clocks are in there because Horvath's a part of it, but like, what are you guys thinking about in terms of pulling together biomarkers, and any that you can mention?
Oh, of course, yeah. Well, the initiative of the Biomarkers of Aging Consortium was put together by a postdoc actually in my lab. His name is Madi Mokri, and he's really the heavy lifter and the spearheader of this whole initiative. And the idea came all about, and he's actually a shared postdoc between my lab and Vadim Gladishev's lab, who's also a PI that's working in the space of biomarkers. The idea came all about by considering the fact that there is a lot of different clocks out there. I think there's now more than 20, just about the methylation clocks. But there is a number of additional clocks that are being developed: transcriptional clocks, metabolomic clocks, proteomic clocks, and so on and so forth. So there is a lot of interest in the field by many investigators to really develop new quantitative methods to really assess in a very kind of tangible and measurable way what aging means on the one hand, but also in particular now that we are, I think, at an inflection point, how rejuvenative interventions, for example, any kind of rejuvenative intervention from exercise to diet to caloric restriction or metformin or epigenetic reprogramming of aging. So what is the impact of any of those interventions on the biological age? So what do they do to the biological age of an individual? And so we came to the idea that, well, again, there is a lot of different clocks, and they're all absolutely, well, not all of them, some more than others, but they're definitely all very useful tools to measure aging at the cellular level. But they're not necessarily always good in capturing the effect of regenerative interventions, right? So, and we have demonstrated, so we have a paper now, we have a study under revision, and we have seen that if you utilize different regenerative interventions and you utilize the same clocks, you see that there is, those interventions are actually acting on the epigenome in different ways. So some interventions affect some parts of the genome, and some other interventions affect some other part of the genome. So, but the question is, what does that mean from a clinical standpoint? Does that really translate into enhanced health span, enhanced lifespan? So what does that mean? So to make a long story short, again, we really realized that, well, first of all, a lot of clocks, different clocks, and there was not a unifying consensus about which of those should be primarily used. Should they be all combined into one super clock of some sort? So, and in particular, like, I think there is really right now the need to also kind of inform the FDA and kind of really make them aware that, well, sooner or later, these interventions are going to become practice in somehow. So how do you measure effectively and quantitatively the effect of any of those on the biological age of people? So yeah, that's the spirit behind the initiative, and the consortium is doing a great work in this regard, and is trying to put together right now, like, all the people that are involved in this field to try to come up to a consensus. We published a paper in Cell, a review in Cell very recently, to kind of, first of all, come up with a consensus language about what aging means, what rejuvenation is.
Yes.
Because we still lack, kind of a common ground, and different people think about this in different ways. So.
Totally. Like, what is aging? And I know scientists get in a flutter when you say you've reversed it, like just using that term. And I think that, I think Matt Kaberlin was sort of a big advocate for, I could be mistaken, so I don't want to, but there are, that we haven't reversed. Certainly there are some scientists who come from that, but then, what are you doing in your lab? I'm assuming this is something that you're going to be grappling with, defining these granular terms and then measuring. But we also know, let me ask you this, that the clocks, so I have two questions for you regarding the clocks. Clocks can behave differently depending on tissue type. So biological age, as you talked about in your own research, what's happening in the brain versus what's happening in, an immune cell, et cetera, that can vary. Are the clocks surrogate markers of what's happening, or are they actually capturing the phenomena, the aging phenomena? Are they just reflective, or
So you're saying, in other words, are those changes that we see with the clocks correlate or causative of aging? So, well, the clocks, as I said, the clocks are very, very useful tools actually to measure aging today. Okay, but they have a couple of fundamental limitations, and that's, I think, the reason why we need to develop new types of clocks, or probably not clocks. We have one, we call it index. Well, first of all, the clocks are pretty much algorithms that are trained on a specific cell type. Okay, and they're trained not only on a specific cell type, but they're also trained on how that specific cell type ages over time.
Yeah. Okay.
And so what they do in simple words is that they look, they try to extrapolate from those cell types, from the methylation of those cell types. They extrapolate through machine learning algorithms a subset of bases in the DNA that change, meaning that they either get hypermethylated over time or hypomethylated over time. And so basically they extrapolate that information, and at the end of the day, what they do is just, they really end up looking at only, in the best case, only maybe at 400 base pairs or so.
Mm-hmm.
In the worst case, just a handful of base pairs. And so basically what they do is just, it's like, trying to simplify a very complex image and try to really kind of zoom in into a very tiny little pixel of that image. And that pixel basically is good in the sense that, it really gives you a remarkable and reproducible way to say, oh, okay, this cell type is age X, and this other cell, the same cell type, from an older individual is now age Y. But the problem is that, well, it needs to be trained on a specific cell type. And so that means that when you use the same clock in other cell types, it may not work as well.
Sure.
Because again, it's been trained on that cell type. So that's the first limitation. And that's the reason why there's multiple clocks now that are being developed. The second limitation is that I think it gives, despite being very helpful, it's looking at such a tiny little fraction of the information, of the whole information, that is really giving you a very kind of blurred image, or of the whole picture, right? It's like looking at, you're in a museum, right? You're in front of a portrait, and instead of looking at the whole portrait, you're just looking at one tiny little pixel of it.
Right.
It can be very informative, but it doesn't give you the whole
That's
complexity
amazing.
of the. And so that's the reason why I think we really need to develop new clocks, because and the third limitation that I should mention is that since they have been built on aging, it doesn't necessarily mean that now with rejuvenative intervention, we're going to see a reversion of that clock in the same, in the reverse orientation. Why? Because the different rejuvenative interventions may be doing different things,
dancing around elsewhere.
And those parts of the genome that are changing may not be necessarily captured by the clock that has been built for a different purpose. And
so this could be why that calorie study that came out, it didn't move any of the clocks. It only moved the pace. So that's it. It's not that those clocks are limited.
No, it doesn't mean that it's not doing anything to the genome. It's actually doing a lot, but the clocks cannot capture that information
right.
because they have been built for a different reason.
Right. So we don't want to throw that intervention out by a long shot.
Exactly.
Fascinating. Okay. What clocks are you using? Like, what do you find work in your lab?
Yeah, so we in our study originally we used the original the Horvath's clock. We actually used two different ones, and in both cases we saw rejuvenation.
Yeah,
but that's an interesting question, Cara, because actually when we start digging, and we did see rejuvenation, by X number of years. But that's an interesting question, because when we start digging a little bit deeper into the data, and we ask ourselves, to go back to your question, like, are these changes correlate of aging or are they causative of aging? So can those changes in the clocks really tell us something about the biology of the cells, or they just change with age, but they have nothing to do with it,
Yes, right.
So we started digging into that, and we saw that the rejuvenation effect that we were seeing with those clocks were explained in both of the cell types that we looked at just by one single base pair in the genome. And that's incredible, because, I was expecting, again, really changes across the whole genome. And what the clock was saying was that the rejuvenation that I saw in my cells was due to the fact that one base pair was changing, and that change was so heavy in the clock.
Wow.
And I said, well, okay,
like
that's
just
great.
One methylation site, like literally a single
so
red lollipop.
that doesn't mean that there were thousands, if not hundreds of thousands of changes happening in the cells, but the clock was capturing only that one. Okay. And I said, well, this is on the one side, on the one hand side, great and exciting, but on the other hand, is like, I need to understand what is going on during this process. I need to understand the logic of this process. I cannot explain the whole rejuvenation by one base out of 7 billion base pairs of the genome that I'm looking at. I mean, that's just impossible. So that's the reason why we started developing new clocks that are looking at the whole genome now.
Wow.
And now in an unbiased way, we are trying to understand if there are features of the genome that are changing in a predictable way as we age, and also as we intervene with.
So you guys are in the middle of creating clocks.
Yeah.
Because you need to actually measure the rejuvenation that you're seeing.
Exactly. Yeah.
That's ridiculously interesting. Are you using like the whole methylome or the whole genome?
Yeah. We are using the whole genome, and we are not using algorithms. So we're not training.
Wow.
We're not training the system in any way. We are just looking at the genome, and we are extrapolating from the genome features through machine learning, features that are invisible to our human eyes, but that a machine learning kind of approach can actually extrapolate, because it can look combinatorially at a number of these, but it's not trained. It's just extracting information from a screenshot, if you wish.
Yeah.
Screenshot taken at a different time. So it's extrapolating the whole information, and it's trying to kind of make an order out of this mass, and try to really come out with features that are biological features that are changing. And to make just to make an example, we found that there is one feature that seems to be consistently changing over time across different cell types. So this can potentially lead to a universal clock, which is a particular part of the genome that is bound by a protein complex. And we saw that this particular part of the genome keep being hypermethylated with time. And when we come in with rejuvenation, for example, with the epigenetic reprogramming, we see that same feature now is becoming younger. So it's becoming demethylated with reprogramming. So
what's that protein about?
So this is a protein complex that is called the PRC2. And excuse me, it's a complex that basically, generally speaking, keeps some developmental genes off in cells. So those are genes that are normally expressed when we embryonically develop, but at some point they need to be turned off.
Right.
We see that these regions that are bound by this repressive complex become hypermethylated with time. And this is associated with an aberrant expression of those developmental genes that now start becoming expressed as we age.
Interesting.
And we don't know if the binding of that complex now becomes loose, and so that's the reason why these genes now are being turned off. Or maybe it's because there's something even at a higher order that is happening. And so maybe these genes not only are bound by this repressive complex, but they're also in the nucleus. They are in parts of the nucleus where they're also being kept silenced. And by hypermethylating them, maybe they detach from those regions and they move into regions of the nucleus where they become active now. So we're trying to understand the biology of this phenomenon, and I think this is more interesting because at least we can start understanding the whole process. Yeah.
It's incredibly complex.
It is.
It's like that joke about epigenetics. Just say it's epigenetics. I mean, it's just so complex. It's really interesting. But it seems like what you're suggesting is that it's possible that later in life, there may be aberrant expressions of genes that should be repressed, that because we're only using them in embryogenesis. But as things maybe get sloppy, or if it's not just sloppy, it's maybe some program phenomena that's happening that it's allowing these to sort of relocate and turn back on. Am I getting it?
Absolutely, you're right. Yeah,
that's interesting. It's really interesting. Okay, so my next question is, you said to me on the podcast that you really thought that like root cause aging, if you will, is happening in the epigenome. But you say it's your opinion. And I just thought that that's fascinating that you're saying it's your opinion, and yet in your research, you're really demonstrating that. So why not put that stake in the ground a little bit more strongly? What's the, yeah.
I think that to clarify what I said the other time was,
Yeah.
I don't necessarily, so there are a lot of different theories about aging, right?
Yes.
There is the disposable soma theory, there is the accumulation of damage theory, and
Yes.
and others. So what I wanted to clarify is that I am agnostic about what the theory of aging is. Okay, I don't know, there's not a consensus as of yet of what's really the root cause of aging. And I don't have a particular opinion about what is the most accurate theory of, they're all inaccurate, but I don't know what is the most accurate of all of them. So that's why I don't necessarily think that epigenetic changes are the causes of aging. Okay, in that sense, I don't know if that's clear. But what I'm really strong and adamant about is that whatever the theory of aging is, again, it can be developmental, it can be accumulation of damage, whatever that is, it results in epigenetic changes that are, in my opinion, and I'm really strong about this, are across all the hallmarks, are the hierarchically dominant hallmark of aging. Okay, so that's my strong opinion. And
it's what you've demonstrated, though, wouldn't you say? Yeah.
And since the epigenome is hierarchically dominant and also reprogrammable, I think that by reprogramming the epigenome, we can, as a cascade, reprogram all the other hallmarks of aging, because they're just, in a kind of a second layer of importance.
Got it. So regardless of the why, this appears to be the business end of, in terms of manipulating all of the hallmarks. Okay, cool. You had talked to me about purposely putting your attention on human cells, and that being the more challenging, but closer to being translatable than than animal models. And we were curious about why that is. What is it about the human cell studies that make it so much harder than animals?
So let me start by saying that I come from basic biology history. As much as I value the animal models, across the spectrum, from the simplest organism to the most complex organism. As much as I value those models, I think they are fantastic ways actually to expand and extend our understanding and our knowledge. When it comes to develop therapies, I think that those models are largely insufficient for the goal that we have. In particular, if we want to have a goal that is in the short term, or not in such a long term. Let's What am I trying to say? In a non-diplomatic way, what I'm trying to say is that I am a little bit sick of seeing potentially groundbreaking technologies and discoveries that work beautifully in animals, and that almost all of the times don't work in humans, because humans are so similar but also so different from animal models. And so I think that now the technology really allows us to start asking the right questions, using the right tools in the right models. For example, using human organoids, or human tissues, or some sort of, again, simplified version of the human organs and the human tissues. And I think I would rather spend a little bit more attention and time and efforts and resources on that, because I think that as imperfect as they are, like the animal models, but they're definitely a step closer to the complexity of the goal that you have in mind, which is treating human diseases and treating human aging. Okay.
Yeah.
And so that's the reason why, I mean, but it's difficult, of course, because of course you have to rely on specimens that are often difficult to get, in some cases ethically concerning also to get, sometimes impossible, because maybe you have to rely on tissues that are not available, and so it's very challenging. But I think it's as challenging as the goal that we have in mind. And I have spent all the entire research that we did on aging was almost entirely done on human cells, because like at the end I wanted to have something that was really that could really have a potential for treating human diseases and human aging. Yeah, I don't know. It was a long way to answer your question.
Well, let me just ask you this, and I know this is not your wheelhouse, and it feels almost like a dumb question, but whatever. As a clinician, we're always trying to translate research. As a clinician in functional medicine, we're thinking about translating research. And actually, since science is sort of in the popular media now, especially longevity, and everybody's like hungry for what's happening, people will extrapolate from animal studies and start experimenting on themselves. But in when I think about it, and my colleagues and I think about it, I mean, if you were going to translate, which I don't know that you would, but if you were going to make decisions on clinical decisions around cell studies versus animal studies, what would they be? Would you look at animal studies, or would you look at cells?
Well, the animal studies are necessary, absolutely.
Yeah.
But what I'm trying to say is that the discovery, in my opinion, now that we have the tools and the technology, the discovery now should be done on human cells and human tissues. And then, of course, it needs to be reverse translated in a way to animals, because you need to do safety, some sort of efficacy also. But the biology of the aging, despite there are a lot of similarities, of course. Our aging is in a way is very similar to the aging that you see in rodents.
Any mammal, yeah.
But it's also so dramatically different. I mean, it happens over the lifespan, which is 80, 90 years old versus 2 years old. Yeah. And it's not just a matter of accelerating a process that is the same. It's not just as simple as that. It's not just a matter of extending that's a biological process over the course. No, that's not as simple as that. Yeah. The complexity, the structural complexity of the tissue is completely different. The cells, the cell types, they're similar, but at the same time they behave differently. The brain, I mean, just how can you study the brain aging, since our brain is so different from any brain of rodents? I mean, I don't think it's a fair comparison. And again, up to now, or up to a few years ago, we didn't have the right tools. Fine, you have to work with what you have, and you have to try to make the best out of it. But now that we have access to cells, now that we have ways to culture human organoids, we can make iPSCs, we can build all sorts of quasi-perfect tissues out of those cells. I think we should start working on those, and this would accelerate dramatically the pace at which not only you discover drugs, but you also find candidates that really have a potential to be translated because they have been tested on human tissues and human cells.
That's right. I get it. Yeah. And so let me just ask you quickly, going back to the clock conversation, the mammalian clock that Horvath recently published on, that's still the pixel, right? You're still looking at a couple of pixels, even though it's across mammals.
Yeah.
Okay. Now, just leapfrogging off of what you just said, about the commitment to human organoids. Where are we at in the clinical trial journey?
So I can speak for what Turn is doing. We are not too far actually from the clinical trial. We just had a very exciting first conversation with the FDA. We submitted an preliminary interact package, which is the very, very, very early conversation. You know, we had some questions about our mRNAs, the cargo, the lipid nanoparticles, and so on and so forth.
Yeah.
And it was great, actually, because we got an excellent feedback from the FDA. And so we had a press release last week on this. I can
Yes, please. Yeah, I'd love to see it. That's
and to the point that actually we didn't really have to have a meeting with the FDA officers. So now our next stage now is really to develop the preclinical studies and then submit a pre-IND package and an IND package after that. So we are really accelerating it now dramatically, because with this first conversation was absolutely important for us to understand how the FDA is thinking about this, because it's completely new territory for them. And it was very reassuring and pleasing to see that actually we are on the same page. And so now it's just a matter of really start developing this technology and to bring it to phase 1. So if everything goes well, money, funding, and so on, I think we are really looking at a phase 1 that would start relatively soon. So hopefully early 2025.
Wow, that's incredible. Yeah, I would love to see the press release.
Sure. Yeah.
I guess I don't know. You know, I don't know that you'll. This is a little off to the right, but Charles Brenner is huge on Twitter, and he's always actually accosting David Sinclair. It's like a kind of a humorous drama that those guys have going, or he has. But he really challenges the publications, Sinclair's publications out of Harvard, and does not think that that the aging phenomena is actually happening and they're reversing it. And I just wondered if you've been following that at all. It seems like you're aware of it, and what your thoughts are on this like sort of heated drama.
Well, yeah, I'm aware of it. I mean, I'm not following it, super closely. Of course, when it comes to science, like in any other discipline, I mean, every time there is something new, this obviously stirs a lot of kind of egos.
Yeah.
So any new idea comes, of course, with the potential, but also with a lot of controversy around it, because until it really gets either disproved or understood, which are, I think, the two sides of the same coin in a way. So I think, I mean, he has a point when it comes to, for example, saying, well, is that paper really the proof that's the fundamental cause? Or to go back to our question initially, like to really show that that's really the fundamental cause of aging, I think is right, in the sense that it doesn't really necessarily prove that that's really the fundamental cause of aging. Also because, again, the method that has been used is through induction of double-strand breaks, which are obviously also impacting the sequence of the genes. So again, I think it's more a theoretical kind of controversy about the theory that explains aging, but I don't think I think he has a lot of reasons to really say that maybe it hasn't been shown that really that's the cause of aging. But again, I don't think that changes the whole picture, because again, it may not be the cause, but it's the manifestation that is really what really we care about, when it comes to rejuvenation and resetting that program back to a youthful state.
Yeah.
So yeah.
Okay, awesome. Thank you. All right. And so then my final question, again, it might be a little bit outside your wheelhouse, but I'm just wondering if you have any thoughts. So for me, being in functional medicine and thinking about nutrition, I was, Randy Girdle has kind of a godlike stature in my field, and the fact that he showed using methyl donors, this remarkable phenotype change in the agouti mouse model. And I'm just wondering, let me see.
for me, this suggests that nutrients are potentially an exceptionally powerful influence on the methylome, on the epigenome, especially when delivered during certain developmental windows. But, we know we can change, from my diet and lifestyle interventions that we could change gene expression. I'm just like, do you have thoughts on that at all? It's maybe a selfish question for me because it's my area, but yeah.
Well, yeah, I do have thoughts in the sense that I absolutely think and firmly believe that nutrition, together with many other things, has a really fundamental impact on gene expression, and it has implications on gene expression again because it has implications on the epigenome. So I absolutely think that yes, nutrients can epigenetically impact our body, our cells, our tissues, and by doing so, they can either accelerate aging or kind of slow it down over time. I'm totally not only the nutrients, but also again caloric restriction, for example, exercise. Even the amount of food,
yes,
is and we in our study on the index we saw that caloric restriction, like epigenetic reprogramming, have the same impact on the same regions of the genome.
Wow,
so they are working through the same mechanism of rejuvenation, if you wish, in ways that again we don't understand. But it's exciting to see that that's happening.
Have you shown anything else to work similarly to epigenetic rejuvenation besides caloric restriction?
No, but we saw that rapamycin treatment, for example, is not acting on the same region. It's working on different regions, and we don't understand why. Metformin, not yet, because we didn't have the data to look at. We were trying to mine existing data sets, and there was nothing available. So far, caloric restriction and epigenetic reprogram seem to be working, at least in part, through the same mechanism.
Yeah, that's super fascinating.
Yeah.
What is this index clock that you're using?
So we call it the PRC2 index, and we have a publication under review now, but it's also publicly available. I can send that to you, so maybe you can read it.
Yes, I would like to. Is this a clock specific for your research in your models, or is this something that you could use, that people could use?
Anybody could use it. Yeah.
Is it available? Or you have the CPGs, the clock structure is available, so you could run it on like the Illumina or something?
Yeah, well, the sequencing can be done on any platform. It can be done, on Illumina. It can be done through microarray. It can be done through any platform. And as I said, it doesn't require any specific algorithm, so there is no code associated with it. So it's just a feature of the genome that you can extrapolate easily without any complex, bioinformatic tool. So it's much easier to look at because again, it doesn't require machine learning training. It's just focusing and zooming into specific regions of the genome that have a specific behavior over time. Yeah.
So it wouldn't really be appropriate outside of a research setting.
No, it would. I think it would, but again, we go back to the question, like what of this of the clocks actually, because the downside of our approach is that you need to look at the whole genome.
Yes.
So it's more expensive than a clock, and so that becomes a little bit more challenging, of course, when it comes to kind of can you use it for every single person? Well, maybe not, because it's very expensive, and so is it worth using it? So that's why I think we need to develop a kind of really consensus about this. But on the flip side, the sequences is becoming so cheap now that with few $100 you can have the information across the whole genome. And so instead of spending maybe a couple of dollars, you spend a couple of $100, and you get a much more comprehensive information about the whole process.
Okay, so that paper and then the press release, if you could send those to me. And that's all I've got, Vittorio. Thank you so much for making some more time.
Pleasure.
Okay.
Bye,
ciao. Bye.
Ciao.
As always, thank you for listening to New Frontiers in Functional Medicine, where because of my sponsors, I am able to bring you the best minds in functional medicine. And of course, today is no exception. Not everybody can be a sponsor on my platform, so I appreciate the good work, the relentless research, and the generous support from my friends at Rupa Health, Biotics Research, and Integrative Therapeutics. These are brands I know and trust in my own clinic, and I can confidently recommend them to you. Visit them at rupahealth.com, bioticsresearch.com, and integrativepro.com. And please let them know that you learned about them on New Frontiers. And if it's not too much to ask, I would really appreciate a thumbs up or a kind review wherever you're listening to New Frontiers. Thanks.